Hello friends, warm welcome to all to our channel World of Zoology. This is our fourth lecture on the topic application of biotechnology, and in that we'll consider fourth application, that is green genes, that is green fluorescent protein GFP from jellyfish Aquaria Victoria. Green genes, green fluorescent protein from jellyfish, which serves as a valuable as a reporter genes used to detect for food poisoning. The aquatic environment, especially marine environment, serves as the rich source of the biotechnological application. One such example is of novel gene from bioluminescent jellyfish Aquaria Victoria. Aquaria Victoria can fluorescence and glow in dark because of the gene that codes for the protein called green fluorescent protein. Actually, this organism, Aquaria Victoria, also sometimes called as the crystal jelly, is bioluminescent hydrogen jellyfish, or which is hydromedusa. It is found off the west of the North America, and this species is best known as the source of two proteins involved in bioluminescence mechanism. The luciferin, which serves as the substrate, it reacts with the oxygen in presence of the enzyme luciferase so as to emit light, and therefore these two proteins which are involved in the bioluminescence, one is aquarin, that is a photoprotein, and another green fluorescent protein which is also known as GFP. GFP is responsible for the production of bright green glow by these jellyfish when exposed to ultraviolet light. This green fluorescent protein, due to its property of fluorescence, can be used to create a reporter gene construct. Here the Aquaria Victoria is shown in this slide. The reporter genes are so called as these can be used to detect expression of genes of the interest in biological system. Hence here, reporter gene is responsible for reporting the presence of the gene of the interest. The green fluorescent protein gene is ligated to promote a region of a gene of interest in order to construct the reporter gene plasmid. These are then introduced into the required cell type. These plasmids, after getting into such a host cell, they are able to transcribe and then translate to produce fusion protein, which has the property to fluorescence when exposed to UV light. These plasmids here serves to detect the report the expression of the gene of interest. Means plasmid with GFP green fluorescent gene serves to detect or report the expression of the gene of the particular interest. Martin Chalfe, Osamu Shimomura, and Roger Saini were awarded 2008 Nobel Prize in Chemistry for their discovery and development of the green fluorescent protein. This approach has found a wide application in basic study of the process of the gene expression and the regulation of the gene. Green fluorescent protein expressing embryonic stem cells are used to track their differentiation into different cell types during development of the embryos. Here you must remember our previous lecture in which role of the embryonic stem cell is described by me during that lecture. That means embryonic stem cells, these cells possess capacity to develop into any type of or any body cell. And here GFP is used to track their origin and differentiation of embryonic stem cells into different cell types in the body of the developing embryos. GFP reporter gene constructs have been innovatively used in the advances of the medical diagnostics and disease treatment. That is for the diagnostic purpose and also for the treatment of the particular disease. They have been used as marker in the tumor formation in mice to follow the progress of the bacterial infections in the intestine, to monitor the death of the bacteria following the antibiotic treatment which is given, and also to study the presence of the food containing microorganisms in the human digestive tract. These GFP serves as the marker gene here. Use of GFP in detecting food poisoning due to Campylobacter jejuni infecting human intestinal cells have been an effective medical diagnostic technique. Reporter genes can also be used for constructing whole cell biosensors as specific and sensitive devices for measuring biologically relevant concentrations of the heavy metals which are the pollutants in the environment. The genetically engineered E. coli bacteria having green fluorescent protein reporter gene produce fluorescence as a measurable signal when the bacteria are in contact with bioavailable metal ions such as cadmium. Such bacterial biosensors are useful and applicable in detecting the availability of the heavy metals in soil wastewater. Architecture of the Aquaria Victoria green fluorescent protein is shown here in this diagram. And with this, we have finished the topic application of biotechnology in our lecture. Here, the last application is green fluorescent protein from the jellyfish Aquaria Victoria, which is shown in this diagram with the in part which is highlighted of the green protein, that is green fluorescent protein from the marginal tentacle of the animal Aquaria Victoria, which is also shown in this diagram or in this slide. The four applications of the animal biotechnology: first is the DNA fingerprinting, then second one, that is recombinant DNA, use of recombinant DNA technology for the production of the medicines such as recombinant insulin. Then gene therapy, again which is divided into two types according to the modality. One is the ex vivo gene therapy and another is in vivo gene therapy with the example of the two skid and cystic fibrosis. Then green genes, which is our last application of this lecture or the discussion for the today's lecture. Thank you for watching this video. If you like the video, do like, share the video for the benefit of the maximum students, and subscribe channel World of Zoology for notification of my next lecture. Thank you once again.